It's my pleasure to introduce Steve Henikoff, Principal Investigator and member of the Division of Basic Sciences at the Fred Hutchinson Cancer Research Center. His talk will be titled Genome-wide Mapping of Protein-DNA Interaction Dynamics. Please welcome Steve.
Okay, I'd first like to thank the organizers for the invitation. Now, I'll be talking about protein-DNA interaction dynamics, but what do I mean by that? Well, all of the active processes that take place in our genome, including replication, transcription, repair, whatever, have to take place in the context of chromatin. That is, they have to deal with the fact that chromatin consists largely of nucleosomes where the DNA is tightly wrapped around, and so those nucleosomes cores act as barriers. And we've been interested in these interactions, and over the years we found that the methods that are available for studying these interactions have limitations, and sometimes it leads us to develop some new methods. What I'd like to tell you about first is a strategy fairly new that we've introduced. I'll introduce you to it, and then I'm going to show you how we can use it to look at a dynamic process, in particular, how a transcription factor can find its site in the context of chromatin. Now, when you think of mapping the chromatin landscape, what you'll probably think of is chromatin immunoprecipitation. It's been around since like 1985, where you cross-link and then shear up the DNA, and then you add an antibody to immunoprecipitate and then purify the DNA. And over the past dozen or more years, the preferred readout has been DNA sequencing, but the basic method really hasn't changed very much. Now, chromatin immunoprecipitation is not the only way that not the only method that you can use to map the chromatin landscape. For example, there's methods that we refer to as enzyme tethering methods. And an early such method was introduced by Bas van Steensel, in which we made a fusion protein between E. coli deamethylase and our chromatin protein of interest. And deamethylase will methylate the adenines of GATC sites, so it'll methylate those sites in its vicinity, and then it's a matter of mapping where the methyls are. Then a few years later, Uli Lemley substituted micrococcal nuclease for the MNase. So micrococcal nuclease will get base pair resolution, as you'll see. And he called this check, or for chromatin endogenous cleavage. And Gabe Zentner, when he was a postdoc in the lab, turned this from a Southern blot readout into a modern sequencing-based readout, a check-seq. However, it's this method CHIC that we've really paid the most attention to. And our assay for this, and this was developed by Pete Skeen and myself, that we call CUT&RUN. It goes like this. We mix live cells with lectin-coated magnetic beads. They stick. We permeabilize, and at the same time we add an antibody. The antibody will diffuse in to find its target, say a transcription factor. We then add a fusion protein between protein A and MNase. Now, protein A will bind to the antibody and anchor the MNase such that when we activate the enzyme with calcium and it cleaves on both sides, it releases the fragment, and we simply allow it to diffuse out into the supernatant. We extract the DNA and do paired end sequencing. From live cells, purified DNA takes about a day, and the backgrounds are exceptionally low. The reason for that is that, unlike CHIP, where you start out by solubilizing the entire cellular contents and then add an antibody, pull a needle out of a haystack, with CUT&RUN, we leave the haystack behind. So here's an example where we're looking using the same monoclonal antibody that was used by the ENCODE project using CHIP for H3K27 trimethylation, which recognizes large polycomb silence domains. And what you see is that if we downsample, so we have the same number of reads for ChIP-seq versus CUT&RUN, you can see that you get better feature definition, and that's because of this low background from sampling from the entire genome for CHIP. CUT&RUN is also very sensitive, so in this experiment we can go down to 100 cells without any loss of data quality at this level of resolution. And then we can do this with transcription factors. This is work of Pete And what you see here is that if we map the ends of the fragments to get a footprint, you can see that the protected region by CTCF is exactly 44 base pairs. And we see that over 2 orders of magnitude range of digestion. What we think is going on here is that over the course of digestion, we're doing a low temperature to slow it down. Over the course of digestion, more and more of the CTCF sites at any particular position will get released until we get limit digestion. So we think it's actually quantitative. Now, because of the nice features of this method, the low background means that you don't have to sequence very deeply, means that it could be quite economical. And Derek Janssens, who did the robotics on this, hooked up with Jay Sarthy, who was interested, a pediatric oncology fellow, who's interested in actually applying this for patient samples. And as proof of concept, we did it on patient-derived xenografts. So what you're looking at here are flash frozen and thawed brain tumors from mouse, ultimately derived from 2 different patients. And they get disaggregated, we mix with the beads, we array in 96-well format, and we run the whole thing on a robot and carry out all the cut-and-run reactions up to the addition of barcoded adapters. We then take it off, do PCR, put it back on the robot for a cleanup, do some QC, mix up the 48 samples, do paired-end sequencing. And when we price this out, our in-house costs are about $50 for 6 million mapped human fragments per sample. And that actually compares very favorably with what people have been doing for profiling, for example, whole exome sequencing. Could cost 10 times as much by commercial rates. And there you get several hundred mutations, but here we actually get the entire epigenome landscape, so to speak. So if we use an antibody to H3K4 dimethylation, which gets the promoters and enhancers throughout the genome, and we look at replicates here for fresh cells or flash frozen and thawed patient-derived xenografts, you can basically see that we get similar data quality. And you'll also notice that for the 2 different patients, these are pediatric gliomas. They have different driver mutations that are responsible. And so therefore, you got very different epigenomic landscapes because you see features here and here that actually differ between the 2. Now, this particular gene is deleted or mutated in many adult gliomas, but this pediatric glioma with very low mutational burden, and you don't see mutations, but what we do see are we can get, actually, here's a single nucleosome that's over the shared promoter. So this is very informative. And right now at the Fred Hutch, the Genome Shared Resources is offering this as an automated service for the researchers as well as clinicians, and they're already getting going on doing this for patient samples. And I should point out that the methods are easy to do, and so therefore we have a protocols online where you can cite here that Derek runs, that you can ask questions and Derek give you an answer. And there are a lot of people who've gotten this to work. And over the past couple of months, there have been kits that have shown up. Now, it's getting more and more popular. So, for example, if we look at Google Scholar we look at publications, listen to Google Scholar with the word ChIP-seq. You can see last year there were 10,000 such publications. TAG-seq, popular method, beginning to catch up. And CUT&RUN is also on a similar trajectory. So it's getting popular. But meanwhile, we've kind of switching over in the lab to a different method. And this is work of Hatice Kayakur. And here we start out the same as with CUT&RUN and adding the antibody to the permeabilized cells. But instead of adding protein A aminase, we add protein A TN5. Now, TN5 is a cut and paste transposase that was developed by Illumina in their Nextera system, in which the transposase will insert adapters, sequencing adapters on the ends of sequences, so that eliminates the need to make libraries. It's also the basis for the popular TAG-seq method, but here we're tethering it. So we just activate the enzyme with magnesium, we extract the DNA, we amplify, make sequencing libraries. And now this takes 1 day from live cell sequence ready libraries It's really, really kind of slick. We've even removed we've even eliminated the extraction protocol, so it's even easier to do. And the data that we get looks quite nice. So here, for H3K4 dimethylation, we compare CUT&Tag to CUT&RUN, and you can see you get nice feature definition, low background compared with ChIP-seq or a TAC-seq, which is another way of getting some of these regions, the promoters and enhancer sites. Now, a more systematic way of analyzing the data is something that ENCODE introduced called FRIPS, fraction of reads and peaks. The idea is that the higher the fraction of the reads and peaks, the better the signal and noise is. That is, you're comparing the peaks to the background. And what you can see is CUT&Tag has actually even higher signal and noise than CUT&RUN. Now, it turns out that the CUT&Tag is very sensitive to the exact antibody that you use, and we discovered this when we found we got a great antibody for H3K4 trimethylation and increased the FRIPS like an order of magnitude. So this emphasizes the fact that if you have good antibodies, this method can work quite well. So here we're using that same antibody I mentioned before, H3K27 trimethyl, which gives us polycomb silence domains. So here's a nice polycomb silence domains. When we compare to ENCODE, where it requires about 1 million cells or so, and you have to sequence pretty deeply in order to get this kind of profile, you can see with CUT&Tag about 3 million cells and 3 million fragments, 60 cells. This is enough to give you pretty good feature definition here. And this encourages us to take a single cell approach. And we've adopted the Takara iCell8 system, where we carry out all the reactions from antibody binding to integration in the single tube, and then we array it in 72 by 72 format and nanowells, such that on a chip about that big, and then we do barcoding PCR on it. And this is the kind of data that we've been getting. So for 900 single cells, you can see that the aggregate is pretty much similar data quality as what we get in bulk, and these are single cell, single molecule tracks. We can do something like we can mix cells, for example, human ES cells to K562 cells, and we look at the HOXP locus, which is silent in the human ES cells, so it's polycomb marked, and you can see the difference. And if you like clustering, you get nice clusters out of it. So we're currently working on actually applying this to a developing system, which brings me, which I'm going to get to next. So CUT&Tag is now also very popular. It's perhaps more popular than CUT&RUN. We've already sent out about 500 aliquots to 500 labs, and here I answer the questions for the CUT&Tag protocol. Now, okay, so that's a toolkit, and now I want to tell you about how we've been able to use CUT&RUN in order to address an interesting problem. And this is work of Mike Mears, postdoc in the lab. Now, if you think about it, the 70% of the genome is wrapped around nucleosomes pretty tightly. So how does a transcription factor get in there to find its site? And there are two general hypotheses that people have been arguing about. So one is what we might refer to as site exposure, and that is there's transient unwrapping. It might be facilitated by remodelers that use ATP to do that. And the other idea is pioneering, where the transcription factor sits on the surface of the nucleosome, recognize it, and in some way will, over the course of, say, development, will kick it out. And Mike realized he could use CUT&RUN in order to distinguish these models and see what's going on. And to do this, we adopted So we adopted a human ES cell to endoderm system. So SOX2 is a pluripotency factor, and this is the CUT&RUN signal. Okay, I'm going the wrong way. Okay. So what you were looking at here, I don't know why it's not. Do I have to point? Okay. So this is I don't know. Okay, hold it. So I'm just hitting the laser thing. Okay, I'm going to forget the laser here. Is there another pointer? Okay. I didn't hit that black button. Can you put it back on?
Let's get him.
Walk me through his own.
Okay, so I'll go on. So what you're looking at was a heat map where you basically call, so the idea is that for SOX2 cut-and-run data, we basically call peaks and then line up all the peaks and then order them by increasing occupancy of the SOX2, so you get this nice pattern. And what you were looking at was in the ES cells, which are pluripotent, and that's where SOX2 acts, you were seeing that there's that you get a strong signal in day 1. In day 2 of differentiation, the signal pretty much goes away. The differentiation factors, SOX17 and I think FOXA1 maybe, will kick in later. So they'll start out negative, and then they'll just increase over the course of differentiation. And then there's CTCF, which I already mentioned, which is constitutive. Okay, so now let me just try to explain the idea here. Okay, so just imagine that you have, oh great, okay, maybe you better do it. Yeah. Okay, so if you keep going. Yeah, keep going. A few more clicks. Can I actually do that? Okay, so the idea is pretty simple actually. Keep going. Ah, you skipped that. Is it our time?
More or less.
I didn't.
That looks better.
Okay.
Use the mouse, and you can do it right here.
Oh, use the mouse?
Yeah.
Oh,
Just
great.
click back arrow and forward arrow. Yeah.
So somebody time my talk?
Yeah.
Okay.
You'll want to click on the arrow physically, well, with the mouse.
Oh, I'll do it right here. Oh, okay.
There, you just let
I
me.
just
I'll
want to
press it.
get that cartoon. Stop. Okay. Okay, so here's the idea. Okay, a transcription factor sitting on the surface of the nucleosome can't get the DNA that's right around it. It's blocked by the nucleosome, but it can reach around, get the whole nucleosome. So what you'll see is early in differentiation, if it's sitting on the surface of the nucleosome, you're going to get large fragments, and then as it goes out and that nucleosome goes away and it binds to the naked DNA, it'll become a small fragment. And we do this locus by locus. So that's what you'd expect only for a pioneered locus. So next slide. Okay, so is it safe to actually use this?
Use the laser. I'll click forward for you.
Okay. Okay, so CTCF control, and there are not really very many sites that you see show pioneering activity, but next slide. FOXA2 is an example of a pioneer factor, and 29% of the binding sites show this pioneering activity by this assay. Next slide. And so this turns out to be quite general. So for CTCF, can you just hit the next button? Yeah, only a small percentage actually show pioneering activity, but you see all of them, all the differentiation factors show some pioneering activity. GATA4, for example, about 50%. And next slide. Okay, that's fine. Okay, so we haven't used any sequence information. We've only used the cut-and-run data to find the sites. So therefore, we can ask, what's the difference between the pioneered sites and the non-pioneered sites? Is there some difference? And what you see here is that if we consider that the consensus is what the transcription factor has evolved to bind, then any deviation from the consensus would be a lower affinity site. And what we see is for all the pioneer factors, we see that the pioneering sites actually are lower affinity. So let me try to make this intuitive as to why that might be. So the next slide, here's how we're thinking about it. So if it's a high affinity site, non-pioneering site, it'll just bind. However, if it's bound to the surface of the nucleosome, then other parts of the protein can stabilize it. So for example, FOXA2 has 2 winged helix domains. Those are found in H1 that Barbara referred to. And the winged helix domains will sit on the surface of the nucleosome and stabilize it. So there's extra binding energy from that. And so the way we envision it is the factors are actually bouncing from one nucleosome to the next. And when they're in the right orientation, they can test out that binding site and hold on to it long enough. And the longer it holds on, the more likely it is to have its effect on activating the gene. And from an evolutionary context, I think that makes some sense. So if you imagine that the sites to find new sites over the course of evolution, let's say a site that if the factor binds, it'll be selected for. Then if it's a lower affinity site, then there's just going to be more of those than the high affinity sites. That's one thing. And the other thing is that once it actually binds that new site and it gets selected for and goes to fixation in the population, then there will be no selective pressure for it to go to higher affinity. So we do find that transcription factor binding sites have very little information compared to the size of the genome that they have to deal with. And so we think that this may be a general rule for how the transcription factors have evolved. So with that, I'll just close. And I think I talked about the people who have been involved in this work, also Kami Ahmed, who's part of the CUT&Tag project, and others. If you're interested, we're still sending out protein ATN5 aliquots. You can just send an email to that email address, and we'll get you going. And I'll stop there and take questions.
If people would raise their hands, someone will bring around the mic to you, and then we capture the question and the answer.
Hi, very nice talk. Does the cut-and-tag method also work for other antibody for TF rather than like a histone mark? Because it requires higher temperature for reaction. So
no, okay. I'm not sure. So for cut-and-run, cut-and-tag, they're pretty much the same with respect to. So they're pretty much the same with one exception, and that is, it's not so much that the temperature is a problem. What the problem is that we have to go to higher salt when we do the tagmentation, because otherwise we get the accessible sites. So we get the tag-seq sites that are that are superimposed. So that's a problem for transcription factors because salt is really competes for DNA-protein interactions. And so for that reason, we don't really recommend for transcription factors. We still recommend using cut-and-run. It works quite well for that. So mostly we're using this for histone modifications and for the single cell profiling we're using cut-and-tag for.
thanks.
Steve, I was just remembering some data from Steve Tapscott when he was looking at, I think it was MyoD, and he saw many thousands of these sites that I gathered were low affinity, that he had to sort of hope that some of them were high affinity. Will you be able to sort through that kind of?
Yeah, I think so. We haven't, I don't think Steve, I don't know if he's done MyoD with cut-and-run, but I would expect that you would see the difference because we do think that cut-and-run is quantitative for transcription factors.
Yeah, I think,
So
I remember his first ChIP-seq, and it was kind of like, oh no, that's too many sites there. And
Well, so ChIP-seq, because of the cross-linking, can cause epitope masking, is not, we don't think that it's fully quantitative. This is all native, so actually I think it probably is quantitative. And we can do the time course.
questions?
Yeah, Charles.
Microphone.
Steve, how much information is in these binding sites in terms of specificity? You talk about low.
I showed a
affinity.
sequence logo for FOXA, and you
Uh-huh.
can see that it was only about 6, 7 amino acids wide, and they weren't, they were quite variable.
And did I miss how it gets additional specificity to avoid errors?
Well, I think it has a lot to do with, even at the low specificity, the energy of binding of, say, for FOXA2 for the winged helix domains, that energy, it'll add on to that energy. So that's part of it, I think. So it makes it very sensitive to the off rate. So in other words, if it stays on long enough, then it'll have an effect. If it doesn't stay on long enough, it'll go away. Mm-hmm. And I think it gets a boost from the interaction with the rest of the protein. Mm-hmm. But all of these factors have other parts of the protein that can interact with nucleosomes. So they all have some nucleosome binding activity. Mm-hmm. Mm-hmm. So I think for the same factor, it's going to be affected by that. And of course, there are other. That's not the only thing that can give you specificity. I mean, once it binds, then a whole series of processes, including remodeling, can occur. Mm-hmm. that will move it along. Mm-hmm. And along those lines, I should point out that 3 days of development of differentiation is a long time, considering that transcription factors in vivo have dwell times of only about 2 seconds, something like that. So there's got to be more to it than just transient binding.
Mm-hmm. Okay, thank you.
One more question.
I was just wondering, those pioneering sites that FOX2A would bind to, the low affinity sites, once the nucleosomes shifted out the way, within that DNA, is there evidence of motifs for more cell type specific transcription factors binding?
Exactly, and that's what's seen. For example, so at some of the sites, GATA4 might bind first. Eventually, SOC17 will bind sort of sometimes nearby, and we think that they're binding independently, but they are having effect on the same nucleosome, so that could help. So the basic idea is that as the concentration of factors build up, and as the residence time is longer and longer, basically, because there's just more of it, one comes off, another one comes on, and this is happening at multiple sites, eventually that nucleosome will get kicked out. That's the idea.
Great.
Okay.
Please join me in thanking Steve for a great talk.